In the past, what that's meant is you make antibodies to something and it increases the blood clotting. This is brand new. This antibody-dependent enhancement attaches to part of that spike protein called the N-terminal. And when it does it, it opens up the spike protein to make it more infective. And they discovered that part of the reasons why people in hospitals were not doing better even when they made antibodies is because the antibodies they made might have too much antibody to the N-terminal domain. So just because you can make an antibody doesn't mean it's the right thing to do, which raises questions about vaccinations for the number of people to get them to make antibodies. The goal shouldn't be so much to try to prime you for when you get infected, but to treat you when you get infected and to treat the inflammothrombotic response if you get COVID-19. There's a lot of terms used about the efficacy of vaccines, and you should know three terms. The relative risk reduction. This is what you've heard so much about, the 95%, the 94%, the 65%. This is how many people got diagnosed with COVID in the vaccinated group versus the people who got diagnosed with COVID in the non-vaccinated group. There's a more important term for you to understand, and that's absolute risk reduction, which is a comparison of those groups that says, what's the real difference between those groups? And we're going to look at those numbers as well. And then there's the number needed to vaccinate, which is how many people would you have to vaccinate to try to reduce one person from being diagnosed with COVID? These are what you hear about. So what does vaccine efficacy mean? It really means, when they're saying this number, that they're looking at the number of people who got vaccinated and then were diagnosed with COVID. This is one example from Pfizer. Compared to the people who did not get vaccinated who got diagnosed with COVID. And when you do the math, it gives you 0.05. 1 minus that number tells you vaccine efficacy. You multiply it by 100, it gets you percent. So how did they decide who made the diagnosis of COVID? Well, you had to have two things. You had to have a positive PCR test, and you had to have a set of symptoms. Now, Pfizer's group is that group. The symptoms that are there are the same symptoms you would get with any infection. Viral, bacterial, fungal, tuberculosis, and in some cases, cancer. It's not very discriminating. Moderna, same problem, same issue. Again, I know they're small. You can download them, read through them. They're what you would normally have for symptoms. Aches and pains, muscle discomfort, diarrhea, cough, runny nose, that type of thing. Your body's letting you know something's wrong. And Janssen, this is Johnson & Johnson. The company that makes it is Janssen. That's why I refer to it. This is a Belgium company, which, by the way, Belgium pulled this from the market. So, you got those two things, you got a diagnosis of COVID. So let's look at the actual emergency use authorization documents. And if we look at Pfizer's to begin with, and we do what they just asked us to do for vaccine efficacy, you'll find in the confirmed cases that 8 people who got vaccinated were diagnosed with COVID. Okay? In the non-vaccinated group, 162 got diagnosed with COVID. And you do the math, and it comes up with a 95% efficacy. But you're not asking how often you're going to get COVID. Your question is, will it prevent me from getting diagnosed with COVID? Will it prevent me from dying from COVID? So let's look at the data. Their data, not mine. If we ask for confirmed COVID cases, you'll see that in the vaccinated group for Pfizer, there were 17,411 people. There you go. 8 people were diagnosed with COVID, which leaves you with 17,403. It gives you a value. Non-vaccinated, there were 17,511. 162 were diagnosed with COVID, which leaves you with 17,349 who did not develop COVID. Those are the numbers. If you're a scientist like me, you don't just look at those numbers, you statistically analyze them to ask a question, is there any real difference between those two groups? And it turns out that when you do that, there's not. It's not statistically different. But looking at that more important number, what was the absolute risk reduction? Well, remember this? 0.93 minus the difference in those vaccinated gives you an actual absolute risk reduction of 0.88%. If you ask if it cut down on the risk of death, well, here's the data. Page 41, Pfizer had 2 deaths, no vaccine group had 4. And what does this say? There is no statistically significant difference in the numbers of deaths, and they represent what is seen in the general population. So Pfizer, no statistical difference whether you get vaccinated or not, and no change in deaths. Let's look at Moderna and do the same thing. These are the emergency use document presented to the FDA that resulted in their being granted EUA clearance. COVID diagnosis, 11 people with COVID in the vaccinated group. Whoops, back that up. Richard, not so fast. In the non-vaccinated group, 185 over here gives you a vaccine efficacy of 94%. Okay? Same thing we do with Pfizer. Now let's look at the likelihood that it's not going to cause COVID, because that is, after all, the goal. So in the vaccinated group, there were 13,934 people. There they are. 11 ended up with COVID, which leaves you with 13,923. That's the rate. Non-vaccinated group, 13,883. 185 with COVID, therefore 13,698 did not. It is amazing what you can find if you go into the documents and dig, but you have to dig. Is there differences between those groups? No, there isn't. And more importantly, the absolute risk reduction, you saw 1.33% ended up with COVID diagnosis. That's there, minus 0.08% in the vaccinated group, and your efficacy is 1.25%, the absolute risk reduction. Deaths? Well, here they are, 6 in the Moderna group, 7 in the no vaccine group. And as you can see, again, the statement, there's no statistical difference. And it's what you would expect to see in this many people in the general population. This is my favorite vaccine because it gives us a little bit more view into what's really going on. Because they use 14 days, they use moderate and severe as the definition at 14 days, and at 14 days, also 28, but at 14 days, if we look at their data, 176 in the vaccinated group, COVID diagnosis, 513 in the non-vaccinated group. At 14 days, that gives you that efficacy of, thank you. Reading glasses, please. 66%. Okay? 28 days, they also have that data. 114 diagnosed in the vaccine group, 326 in the non-vaccine group, and the efficacy here is 0.65 or 65%, right? All right, those are the numbers reported. Let's tease it apart and look at the information we really want to know, which is the likelihood that you won't develop COVID. All right? So, using the definition of moderate to severe or critical COVID, at 14 days, you'll see 21,636 in the vaccinated group. 176 got vaccinated, leaves you at 21,460. Non-vaccinated group, there were 21,574 people. 513 got vaccinated, which gives you 21,061 who did not come down with COVID and didn't get vaccinated. If you do the math, lo and behold, this is science, there is a statistically significant difference, 0.05. Okay, absolute risk reduction, doing the same thing we did before. There's the risk, 2.38% in the non-vaccinated group at 14 days. 2.38 versus the vaccinated group, 0.81 is a 1.57% absolute risk reduction. Let's go one step more further. Let's go to the 28-day data, moderate severe, because they did, after all, report 14 and 28. And bear in mind, the 14-day data is what they got EUA approval with, not the 28-day data. They stopped at 14. If you go to the 28-day data, there were 21,424 in that group, 114 diagnosed with COVID, leaving you with 21,310. The non-vaccinated group, there were 21,199, 326 diagnosed with COVID, 20,873. No longer statistically different. 2 weeks later, gone. Okay? Absolute risk reduction, again, using these numbers, is 1.54% for the non-vaccinated group minus the vaccinated group, 0.53, is a 1.01% absolute risk reduction. We're going to go farther down the rabbit hole. We're going to go from table 14 to table 15, which also was not used for EUA authorization. Why is that critical? Because every time you do a clinical research study in more than one site, you have everybody making their decisions non-centrally. But everything goes to a central core lab where the people in charge make a decision, yes or no. And it turns out that the results are very interesting when you compare what everybody out there said versus the central lab, which is even mentioned in the EUA document for Janssen. And all you had to do was go one table further in the EUA documents. So EUA non-centrally confirmed, looking at moderate to severe critical COVID, versus the centrally confirmed, which not only included moderate and severe critical, but included the mild cases. At 14 days, there were 176 who were diagnosed with COVID. The central lab at 14 days, that dropped to 117. They didn't make as many diagnoses of COVID. In the placebo group, 513, but in the central core lab, no, there were only 351 who met the definition. At 28 days, non-central, everybody, 114, but the central core lab said nope, nope, 66. 326 in the placebo group, but in the central core lab, no. There were only 195. Those numbers are significantly different as they drop the number of cases by 32 to 42%. Those numbers weren't used to determine whether these drug vaccines should be EUA approved. Here's what really happens when you compare all the things I showed you. In table 14, non-central lab, moderate and severe, these are the numbers. Go to the centrally confirmed, these are the numbers. Go to centrally confirmed and eliminate mild because, let's be honest, you didn't die with mild COVID. These are the numbers. 32 to 42% drop in the cases for individuals with a diagnosis of COVID when you use the centrally confirmed lab results, which was not used to determine whether the FDA would authorize these drug vaccines. Differences in drug deaths. Well, in the Janssen group 5, 20 in the non-vaccinated group. Here's the interesting thing, I think. I'm going to go back on that because I like it the way I want to do it. Of these 5, 2 of them were unknown. Is Dr. Yan available now? So we want compelling evidence of the efficacy and safety of these drugs, and we're testing in larger numbers of people and comparing it with other drugs. Remember I told you there were 10 groups of drugs I had. I was comparing every one of those to every other one, because when we began that trial in April of 2020, there weren't drugs out there. There were just lots of drugs to try. And so instead of doing one drug and spending a year and saying yes or no, I combined them to see if there were combinations, because one thing we learned from those original MERS studies was that viruses and the HIV studies is that you have to use multiple drugs to treat and be successful. You have to target all of those different segments I talked about. So we tested different doses, and when you see, does it need to be used early? Can it be used late? Is there a cutoff to which this drug works? Okay, maybe we can use it for early people, but if they get later on, we have to use another drug. That's how we make these decisions. And once there's enough information, you then go to the FDA and you say, here's our data, we want drug approval. The final 4th phase is after the FDA gives it. It's called pharmacovigilance. This is where you look to see what else. What are the long-term effects of a drug and what else it might be useful? Minoxidil, hair growth, any of a variety of things. This is a slide you wanted to see, that my students would love to see, where I break it down much more simply. Testing animal tissue models. Phase 1, is it safe? Phase 2, is it effective? Phase 3, is it better? But the other steps are what you have to go through scientifically to get there. And you expect us to do that because you expect, if something's FDA approved, that we've done our job so that you don't have to do our job for us. Now, how long does it usually take to develop a vaccine? Typically 10 years. We've done it in 3 to 5, and if you look at all the phases that we just went through, they're color-coded here, overlapping for the shortest duration of time typically gets you to 10 years. This is the emergency use authorization document. You'll notice a recurring theme. It was issued in October 2020. Okay? It's non-binding recommendation, non-binding recommendation, non-binding recommendation, and it doesn't place the FDA or the public under any obligation. These are the emergency use authorization requirements, and the 4th one stipulates that there's no adequate treatment available. That's critical, because if any of these 4 get knocked out, EUA dies. The only way you can maintain EUA is if all 4 of these exist. The problem is there are adequate treatments. So by definition, the EUA doesn't exist. The use of PCR tests is gone. EUA authorized vaccines are gone because the EUA no longer exists. Now, remember this? I showed you this before. These are the 3 drugs, Pfizer, Moderna, and Janssen. These are the different trials, phase 1, 2, and 3 that we talked about. These are the National Clinical Trial website numbers. You can go look them up. They tell you when each of these phases started and when they ended. And last time I looked, none of these 3 drugs are recruiting patients. And they don't need to recruit patients because they've got a slew of them. You're either in the experimental or you're in the control group right now. These are the same color-coded timelines. Every one of these were EUA approved in less than 10 months. And remember I said you have to go phase 1, phase 2, phase 3? Well, Janssen, or Johnson & Johnson, kind of got their dates flipped around. because they started their phase 3 before they started their phase 2. National Clinical Trials Government website.